Box jellyfish, 5 million Gs. That's the acceleration inside a box jellyfish's stinging cell when it fires. Not 5,000, not 50,000. 5 million times the force of gravity, compressed into a structure smaller than the width of a human hair. It is the fastest mechanical event ever recorded in biology, faster than a bullet leaving a rifle barrel, and it happens in 700 nanoseconds, a unit of time so small that light itself only travels about 200 meters before it's over. The animal responsible for this is the box jellyfish, Chironex fleckeri, and almost everything about it contradicts what it should be capable of. It has 24 eyes, not light-sensing patches, not clusters of photoreceptive cells like you find in a starfish. Actual image-forming eyes. They're arranged in clusters of 6 on each of the 4 sides of its bell, called rhopalia, and 4 of those 24 have full corneas, lenses, and retinas. The optical components are structurally similar to human eyes. In every other known organism on Earth, a setup like that requires a brain to interpret the image. The box jellyfish doesn't have a brain. It has a decentralized nerve net, no cortex, no processing center, and yet it navigates through mangrove roots and dock pilings at speeds of up to 7.5 kilometers per hour, actively dodging obstacles, tracking light gradients, and changing course to intercept prey. Unlike 99% of jellyfish species that drift wherever the current takes them, this one hunts. It chooses direction, it accelerates, it pursues. It has eyes that can see attached to a body that, by every neurological standard, shouldn't be able to process what it's looking at. Its tentacles extend up to 3 meters, and each one is lined with billions of those nematocyst cells, each one a loaded spring-coiled harpoon on a molecular hair trigger. The firing mechanism is partially pressure-sensitive and partially chemical. The cell detects both the physical contact and the specific chemical signature of prey skin. Contact with human skin fires thousands of them simultaneously, injecting venom through microscopic tubules that penetrate the epidermis before the pain signal even reaches the brain. The toxin attacks the heart, the nervous system, and the skin cells all at once. The cardiovascular system was never designed to handle that kind of simultaneous assault. A single adult box jellyfish carries enough venom to kill 60 people. In 2021, a 17-year-old boy was swimming in shallow water near Bamaga, a small town at the northern tip of Queensland, Australia. He brushed against tentacles in water barely past his waist. Bystanders pulled him out, performed CPR on the sand, and got his heart beating again. He was airlifted to a hospital. He spent a week in the ICU. He did not survive. His death broke a 15-year streak with no fatalities from box jellyfish in that region. The water that day was calm, clear, and warm, the kind of water that looks like an invitation. But here's where the story turns. In 2019, researchers at the University of Sydney used CRISPR gene editing technology to map exactly how box jellyfish venom kills human cells. What they found was startlingly specific. The venom molecule needs cholesterol in the human cell membrane to latch onto. Without that cholesterol, it can't punch through. It can't kill the cell. And the team discovered that common cholesterol-lowering medications, drugs that have been sitting on pharmacy shelves for decades, when applied to the skin within 15 minutes of a sting, act as a molecular blocker. They stop cell death in its tracks. The antidote to one of the most lethal venoms on the planet was already manufactured, already FDA approved, already available over the counter in some formulations. Nobody had thought to test it because nobody had mapped the kill mechanism at a genetic level. The box jellyfish is 95% water. No bones, no blood, no centralized brain, no lungs. And yet it carries the chemistry to defeat organisms millions of times more complex than itself, delivered through the fastest cellular weapon known to science. Its entire body is a contradiction. The deadliest venom delivery system in the ocean is, structurally, almost nothing at all. It kills between 50 and 100 people a year. Most of them never see what touched them. Irukandji, 1 cubic centimeter. That's the total body volume of an adult Irukandji jellyfish, Carukia barnesi, roughly the size of your pinky fingernail, transparent, nearly invisible in open water, and responsible for a pain response that some medical researchers have classified as possibly the worst ever recorded in a mammal. The syndrome named after it, Irukandji syndrome, is not a single symptom. It is a full-system neurochemical catastrophe. The venom triggers a massive simultaneous dump of adrenaline, noradrenaline, and dopamine into the bloodstream. 3 chemicals that the body normally releases in controlled, measured amounts, flooded into the system all at once. The heart rate spikes violently. Blood pressure surges to levels the circulatory system was not built to sustain. And in the worst cases, the pressure inside the skull rises so fast that blood vessels in the brain rupture. That's how it kills, not by stopping the heart, by exploding the blood vessels inside your head. In 2002, Richard Jordan, a British tourist, was swimming at the Great Barrier Reef off the coast of Hamilton Island. Healthy, fit, 44 years old. He felt what he later described as something like a mosquito bite on his back. He kept swimming. 20 minutes later, the first wave hit. Violent muscle spasms that bent his body involuntarily, followed by excruciating pain in his chest and kidneys that came on so fast he couldn't stay upright in the water. Then came the symptom that survivors describe in almost identical language, the one that makes Irukandji syndrome unlike anything else in toxicology. The absolute, unshakable psychological certainty that the world is ending. Not fear, not panic, a conviction. Patients have begged their doctors to kill them, not from the pain, but from the overwhelming sense that existence itself is collapsing. Researchers still don't fully understand the neurological pathway, but the venom appears to directly alter perception at a level that bypasses rational thought, flooding the brain with a cocktail of catecholamines that distort every signal the body sends. Jordan's blood pressure spiked so catastrophically that he suffered a massive cerebral hemorrhage. He died before the rescue boat reached the mainland. The creature that killed him weighed less than a gram. What makes the Irukandji structurally different from most jellyfish is its arsenal geometry. Most stinging jellyfish carry their nematocysts only on their tentacles. The Irukandji has them on the bell, the main body, as well. Every external surface is armed. There is no safe way to touch it. There is no angle of contact that doesn't result in envenomation. It is a 360-degree weapon the size of a thimble. The pain it causes is consistently rated above kidney stones, above unmedicated childbirth. Some patients who've experienced both Irukandji syndrome and severe trauma injuries have said the sting was worse. Hospital protocols in northern Queensland include intravenous magnesium and fentanyl as first-line treatments, and even those don't always control it. Some patients require medically induced sedation simply because the pain cannot be managed while conscious. And then there's Dr. Jack Barnes. In 1964, Barnes was a physician working in Cairns who had spent years trying to identify the mystery creature behind what locals were calling type A stings. Agonizing, delayed-onset reactions from something nobody could see in the water. He eventually captured a tiny, translucent jellyfish he believed was the culprit, but believing wasn't enough. He needed proof. Clinical, observable proof. So he took a tentacle from the specimen, placed it against his own skin, and waited. Then he placed a tentacle on the skin of a local lifeguard who had volunteered. And then, in a decision that is either the bravest or most reckless act in the history of marine biology, he placed a tentacle on the skin of his own 9-year-old son. All 3 collapsed on the beach in uncontrollable spasms within minutes. All 3 were rushed to the hospital. All 3 ended up in the ICU. The jellyfish was confirmed as the source. It was later named Carukia barnesi in his honor. He tested it on his own child, blue-ringed octopus, total paralysis, full consciousness. That's the signature of a blue-ringed octopus, Hapalochlena, bite. The venom doesn't knock you out. It doesn't cloud your thinking. It shuts down your voluntary muscles, your diaphragm, your limbs, your eyelids, while leaving your mind completely untouched. You are locked inside your own body. You can hear, you can feel, you can think. You simply cannot move, cannot breathe, and cannot tell anyone you're still alive. Medical literature calls it locked-in syndrome. Survivors have described hearing paramedics debate whether they were already dead. The octopus itself weighs about 25 grams. It sits comfortably in the palm of a hand, barely covering the space between your thumb and pinky. And the venom it delivers, Tetrodotoxin, or TTX, is 1,200 times more lethal than cyanide by weight. It works by blocking sodium channels in nerve cell membranes, shutting down the electrical signals that tell muscles to contract. The diaphragm stops. The intercostal muscles between your ribs stop. Breathing ceases, not because the lungs fail, but because the muscles that operate them go silent. A single animal carries enough TTX to suffocate 26 healthy adults. There is no antivenom. There is no antidote. No drug reverses it. The only treatment is mechanical ventilation, a machine breathing for you, until the toxin clears your body on its own. That takes anywhere from 15 to 24 hours. If someone keeps you breathing, you live. If no one does, you die fully aware of every second. But the octopus doesn't produce this venom itself. It outsources. Colonies of symbiotic bacteria, primarily species of Vibrio and Pseudo-Alteromonas, live inside the octopus's salivary glands. These bacteria manufacture the tetrodotoxin. The octopus is, in a sense, a delivery system for something else's chemistry. It provides the housing. The bacteria provide the weapon. This is not a simple parasite relationship. It's cooperative. The bacteria are nourished and protected. The octopus gets a compound so potent that almost nothing in the ocean will challenge it. Most of the time, you'd never know it was dangerous. The blue-ringed octopus spends its life camouflaged as a small beige lump on rocks, coral, or the sandy floor of tidal pools. It looks like a pebble. It looks like nothing. But 1 second before it strikes, it activates specialized pigment cells called chromatophores across its entire body. 60 electric blue rings flash into visibility in rapid, rhythmic pulses, a warning display so vivid it seems almost artificial. By the time you see the rings, the decision has already been made. The warning is not for your benefit. It's a final signal, a biological flare that says the animal has already committed. In 2006, a man at Austinmer Beach, south of Sydney, found a small gray octopus in a tide pool. He picked it up. He placed it on his arm. Someone took a photo. The bite was painless. A microscopic puncture he didn't notice until the blue rings lit up across the animal's skin like a circuit board switching on. Within 10 minutes, he was lying on the sand in total paralysis, fully conscious, unable to speak, blink, or breathe on his own. Paramedics arrived and reportedly discussed among themselves whether he was already gone. He survived only because they made the decision to begin forced mechanical ventilation and maintain it for 24 continuous hours until the tetrodotoxin metabolized out of his system. Had they arrived 4 minutes later or chosen differently, the outcome would have been different. He heard every word they said, and the mothers, the female blue-ringed octopus, doesn't just carry this venom. She weaponizes her own reproduction. Before the eggs are even laid, she injects them with the bacteria-laden saliva, coating each one individually, ensuring the symbiotic colonies of Vibrio and Pseudo-Alteromonas are transferred to the next generation. She doesn't eat during the brooding period. She starves herself for months, guarding and ventilating the clutch until they hatch. And when they finally emerge, each one the size of a single coffee bean, translucent, almost weightless, they are already fully armed, already venomous, already carrying bacteria they didn't choose, producing a toxin they didn't build, capable of killing something thousands of times their size before they've eaten their first meal. The mother dies shortly after they hatch. She gives them everything and then, but not everything that kills in the ocean is invisible. Some of it sits perfectly still on the seafloor, disguised as a shell, waiting for you to reach down and pick it up. Cone snail. They call it the cigarette snail, not because of the shape, because that's how long you have to live after it stings you. 1 cigarette. Light it, smoke it, and by the time it burns down to the filter, you're dead. There is no anti-venom for this animal in any hospital on the planet. None. Not experimental, not in development. None. And here's the thing. The cone snail, Conus geographus, is one of the most beautiful creatures you'll ever see. Its shell is a geometric masterpiece, covered in intricate patterns that look hand-painted. Museums display them. Collectors pay hundreds of dollars for them. Tourists wading through shallow reefs in the Indo-Pacific spot these shells sitting in the sand and think they just found a souvenir. They pick up their own death because they think it's pretty. Underneath that shell is a soft, slow, nearly blind mollusk, one of the biologically slowest creatures on the planet. It has no claws, no speed, no size. It can barely move. And yet it developed the fastest ballistic weapon and most sophisticated neurological chemistry in the entire marine animal kingdom. That gap between what it looks like and what it does is where the danger lives. Here's how it hunts. The cone snail has a modified radular tooth, a single hollow harpoon made of chitin, thin as a needle, loaded inside a long, flexible proboscis. When prey gets close, the snail fires. The tooth impales the fish, injects venom, and once the meal is consumed, the snail discards the used harpoon into the ocean like a spent cartridge. Then it grows a new one. Reloads, chemical ammunition, manufactured on demand. But the venom itself is where this animal becomes something else entirely. A single drop contains over 50,000 different mini-proteins called conotoxins. 50,000. Each one targets a different pathway in the nervous system. Sodium channels, calcium channels, acetylcholine receptors, pain receptors, motor neurons. They attack everything simultaneously from every angle. The reason no anti-venom exists isn't a lack of effort. It's that the cocktail is so absurdly complex, so multi-targeted, that medicine doesn't even know where to begin neutralizing it. You'd need a different antidote for each component. There are 50,000 components. And still, the snail isn't done. Before it even fires the harpoon, it has already won. Researchers discovered that the cone snail releases a cloud of weaponized synthetic insulin into the surrounding water. The fish swims through it, absorbs it through its gills, and within seconds suffers a catastrophic hypoglycemic crash. Blood sugar plummets to zero. The fish loses consciousness, goes limp, drifts in the current, alive but inert. Scientists call this the nirvana cabal. The prey enters a state of chemical bliss, completely sedated, before the harpoon even touches it. The snail doesn't chase its food. It puts its food to sleep first, then walks over and stabs it. A blind, slow mollusk that drugs its prey unconscious with synthetic hormones, then shoots it with a disposable biological syringe loaded with 50,000 simultaneous neurotoxins. In Mauritius, a young diver was walking the shallow reefs at low tide. He spotted a cone snail shell on the sand. Beautiful, geometric, perfect. He picked it up and slipped it into the pocket of his swim trunks, right against his skin. The snail, compressed and threatened, extended its proboscis silently and fired the radular harpoon directly into his thigh. He felt a small prick. Nothing alarming. He kept walking. Minutes later, his leg went numb. Completely. He stumbled, tried to steady himself, and collapsed onto the wet sand. The conotoxins were already cascading through his nervous system, shutting down signals from his brain to his diaphragm, silencing the muscles that draw breath. When his friends found him, he had already died. Silent asphyxiation. No thrashing, no screaming. He simply stopped breathing and didn't notice. The shell was still in his pocket. Inland taipan. 250,000. That's the number of mice a single bite from the inland taipan, Oxyurinus microlepidotus, could kill in laboratory conditions. Extrapolated to humans, one full envenomation is enough to kill over 100 adults. Not 10, not 50, over 100. This is the single most venomous land snake ever tested, and the way it delivers that venom is built for zero margin of error. Most venomous snakes bite once and pull back. The inland taipan doesn't. When it strikes, it commits. Its jaws snap forward in a blur of movement, and it sinks its fangs repeatedly. Up to 8 precise, targeted strikes landing in the same cluster of tissue within a single fraction of a second. Not scattered bites. Not wild swings. 8 rapid-fire injections stacked on top of each other, like a sewing machine punching through fabric. The venom doesn't just enter the body, it floods it. And the moment it's inside, the clock starts running fast. The venom contains an enzyme called hyaluronidase, which acts as a chemical drill. Hyaluronic acid is the glue that holds your connective tissue together, the scaffolding between your cells, the barrier that keeps fluids contained. Hyaluronidase dissolves that scaffolding on contact. It melts the architecture around the bite site and turns solid tissue into fluid highways, giving the neurotoxins an express lane straight to the bloodstream, straight to the organs, straight to the brain. The venom doesn't have to work its way through your body. It carves its own road. The main component riding those highways is a protein called paradoxin. And paradoxin doesn't just block neural signals the way most neurotoxins do. It physically destroys the nerve fibers themselves. Irreparably. The difference matters. A neurotoxin that blocks signals is like cutting a phone line. Reconnect the wire and the call goes through again. Paradoxin doesn't cut the wire. It burns it. If a victim survives, and that's a significant if, they don't simply wake up and recover. Their body has to grow entirely new motor neurons from scratch. Relearn how to move fingers. Relearn how to breathe without a machine. The healing process isn't recovery, it's reconstruction. A single bite extracts around 110 milligrams of venom. In the baked, cracked earth of the Australian outback, prey is scarce. If a rat escapes after being bitten, the taipan may not eat again for weeks. It cannot afford a failed strike. Evolution solved that problem by making each bite roughly 50 times more potent than what's actually needed to kill the target. The overkill isn't aggression, it's economics. When food is rare, you don't leave room for error. And here's where this animal becomes truly strange. The most venomous land snake on Earth, the creature carrying the single deadliest biological weapon of any terrestrial animal, is a coward. The inland taipan is incredibly shy, reclusive, and docile. It lives wedged deep inside cracks in the dry clay soil of Central Australia, in some of the most remote, uninhabited landscape on the continent. It avoids humans with genuine commitment. It retreats. It hides. It flees at the first vibration of a footstep. Since the development of antivenom, the inland taipan has 0 confirmed human fatalities. 0. The most lethal snake in the world has a cleaner record than domestic dogs. In 2012, a 17-year-old named George Van Dorn was bitten on the hand by an inland taipan, kept illegally as a pet. The strike was so surgical, so clean, that he didn't immediately grasp the severity. Within 45 minutes, his nervous system began to collapse. He lost his vision. He lost the ability to swallow. His muscles locked as paradoxin methodically destroyed the neural connections controlling them. He was airlifted by helicopter to Adelaide Hospital and treated with massive doses of antivenom. He survived, but he spent weeks afterward relearning how to move his own fingers, relearning how to breathe on his own. 110 milligrams of venom, enough to kill 100 people, packed inside an animal that wants absolutely nothing to do with any of them. Black mamba. It raises a third of its body straight off the ground, spreads a narrow hood, and opens its mouth wide. And the inside of that mouth is pitch black, ink black, the kind of black that looks like a hole in the air. That's where the name comes from. The black mamba, Dendroaspis polylepis, isn't black on the outside. Its scales are olive, sometimes gunmetal gray. The black is what you see in the last half second before it strikes. The inside of its mouth, gaping open, aimed directly at your face. In parts of sub-Saharan Africa, they call it the kiss of death. Without antivenom, a bite shuts down the lungs in 20 to 45 minutes. The fatality rate in untreated cases is 100%. Not high, not almost, 100%. It only takes 10 to 15 milligrams of venom, 2 small drops, to kill an adult human. But the black mamba doesn't deal in minimum doses. In a single agitated bite, it routinely discharges up to 400 milligrams, enough to kill 20 to 30 people at once. And it rarely bites just once. When cornered, this snake enters a defensive frenzy and delivers up to 12 rapid consecutive strikes on the same target, each one injecting another flood of venom, stacking dose upon dose into the same wound like a lethal sewing machine. This is also the fastest snake on land. It moves at up to 16 kilometers per hour across open ground, fast enough to outpace a jogging human. Most people cannot outrun it, and they don't need to, because the mamba is almost certainly running the other direction. That's the paradox of this animal. The fastest terrestrial snake on Earth uses all of its extraordinary, finely evolved speed to flee. The black mamba is profoundly evasive. It hates confrontation. Given any exit, any gap, any direction to escape, it will take it immediately at full speed. The most feared snake in all of Africa only deploys its lethal fury when a human blocks its last way out. Corner it, and it becomes the deadliest thing on the continent. Give it space, and it vanishes like smoke. The venom itself works through a class of proteins called dendrotoxins, and they operate on a specific and terrifying mechanism. Human nerve cells communicate through channels, tiny gates that open and close to regulate electrical signals. Dendrotoxins jam the potassium channels at the nodes of Ranvier, the relay points along nerve fibers. When those channels can't close, the nerves start firing uncontrollably. Muscles contract and won't release. The body goes into violent convulsions. And then, just as suddenly, the parasympathetic system collapses. The diaphragm locks, and breathing stops. The shift from seizure to silence is fast. In 2012, researchers at the Institute of Molecular Pharmacology in Valbonne, France, isolated 2 specific proteins from the black mamba's venom. They named them mambalgins. And what they found stunned the pharmacological world. In laboratory rodents, mambalgins acted as biological painkillers far more potent than pure medical morphine, blocking pain signals with a precision that synthetic drugs couldn't match. But that wasn't the breakthrough. The breakthrough was that mambalgins produced zero addiction, zero tolerance buildup, zero dependence. The most devastating venom in Africa contained a molecule that could revolutionize chronic pain treatment without the catastrophic side effects of opioids. The kiss of death became a painkiller. Dingo Dinkelman, a famous South African wildlife documentarian, spent his career working face-to-face with the continent's most dangerous reptiles. In 2016, during a rescue operation, he was cornered by a black mamba. The snake moved at arrow speed, delivering multiple rapid strikes that missed him by millimeters before it found its escape and vanished. He survived that encounter. Years later, in 2024, Dingo died after being bitten by a green mamba, a close relative, due to severe anaphylactic shock. Even the greatest experts live on borrowed time with this lineage. The fastest snake alive, carrying enough venom to kill a classroom. Engineered by evolution into a creature that wants nothing more than to run away. And hidden inside that same venom, a compound that could end human suffering without a single side effect. But not all killers need fangs. Not all of them need to bite or sting or even move. Some just need to be touched. Golden poison frog, 2 micrograms. That's the lethal dose. If you could see it, which you can't, it would look like 2 grains of table salt sitting on your fingernail. Touch that amount of batrachotoxin and your heart enters a rhythm it will never leave. The golden poison frog, Phyllobates terribilis, lives in the lowland rainforests of Colombia's Pacific coast in the Chaco region. It's small, roughly 5 centimeters, about the size of a paperclip, and it is, by conservative estimate, the most toxic vertebrate ever documented on Earth. In the jungle, everything wants to hide. Bark-colored moths press flat against tree trunks. Pit vipers coil under dead leaves in patterns that mimic the forest floor. Camouflage is the default survival language of the rainforest. This frog didn't get the memo. It sits on top of dead leaves in broad daylight, bright golden yellow, practically fluorescent against the brown mulch. It hops slowly, unhurried, in full view of every predator in the canopy. That's aposematism, the biological equivalent of diplomatic immunity. Nothing touches it. Nothing has touched it for millions of years. The color is not a warning, it's a fact. Batrachotoxin works by locking sodium channels in your nerve and muscle cells permanently open. Sodium floods in and never stops. Your muscles fire and cannot relax. Your heart contracts and contracts and contracts, not in a normal rhythm, but in a frantic, uncoordinated frenzy, until it simply exhausts itself and stops. There is no antidote, there is no treatment window. There is a window of a few minutes where your body tears itself apart from the inside. The Embera people of the Chaco have known this for centuries. Their hunters catch the frogs using broadleaves, never skin contact, and rub the tips of their blowgun darts slowly across the frog's back. The secretion soaks into the wood, and here's what makes it terrifying as a weapon. That residual toxin on a dried wooden dart stays active and lethal for over a year. When fired through a blowgun in the canopy, the dart doesn't need to hit a vital organ. It doesn't need to penetrate deep. A grazing scratch across the skin of a monkey or a bird is enough. The prey drops from the treetops in instant cardiac arrest before it has time to take another breath. But take this same frog out of the Colombian jungle. Put it in a clean terrarium in a zoo in Denver or Berlin. Feed it common fruit flies and crickets. Within weeks, its toxin levels begin to drop. Within months, it's functionally harmless. You could hold it in your bare hands. The most lethal vertebrate on the planet becomes a pet because the frog doesn't produce batrachotoxin on its own. It harvests it entirely from its diet. Specific species of tiny beetles and ants found only in the Chaco lowlands, insects that themselves accumulate the toxin from plants and fungi in a chain of biochemical handoffs that ends on the frog's golden skin. Remove the diet, remove the death. It's a contract killer. The poison was never its own. And the reason the frog doesn't die from carrying a substance that kills everything it touches? A single amino acid substitution in the structure of its sodium channels. One molecular key changed. That's the distance between carrying death and becoming it. One key, hooded pitohui. In the highland forests of Papua New Guinea, there's a bird the local tribes call the rubbish bird. Not because it's ugly, it's actually striking, black and orange, about the size of a jay. They call it rubbish because no one can eat it. No preparation technique, no boiling method, no amount of cooking makes the hooded pitohui dicrus, safe to put in your mouth. Every family that's tried has learned the same lesson. Some only once. For most of modern science, the idea of a poisonous bird was treated the way you'd treat someone claiming their cat could do algebra. It wasn't taken seriously. Birds were beaks and feathers and migration patterns. Venom belonged to snakes and frogs and jellyfish. Then Jack Dumbacher showed up. In the late 1980s, Dumbacher was a young ornithologist working in Papua New Guinea, netting birds for field research. A pitohui scratched his finger while he was untangling it from the mist net. It was a small cut, a nothing cut, and like anyone with a bleeding finger, he put it in his mouth without thinking. Within seconds, his lips went numb. His tongue tingled aggressively, then lost sensation entirely. A heavy, paralyzing blockage moved through his mouth and into his throat. He didn't die. The dose was microscopic, just feather dust on a shallow scratch. But he stood there in the jungle with a numb face, realizing he'd just become the first documented case of a venomous bird affecting a human being. The toxin in the pitohui's feathers was batrachotoxin, the exact same compound carried by the golden poison frog. On the opposite side of the planet, in a completely unrelated animal, a bird. The pitohui doesn't manufacture the toxin either. It eats choricin beetles, small, metallic-shelled insects found in the forest leaf litter, and processes the chemical through its system, depositing it deliberately across its feathers and skin in what amounts to a full-body chemical baptism. Every feather becomes a surface weapon. Researchers at the Smithsonian extracted toxin from a single feather and tested it on lab mice. The mice died in minutes, convulsions, hind leg paralysis, cardiac arrest. 20 micrograms scraped from the plumage carries enough poison to kill an infant through contact alone. And like the frog, the pitohui carries a genomic mutation that blocks batrachotoxin from binding to its own sodium channels, immune to its own armor. The same evolutionary trick, the same molecular lock, arrived at independently by a frog in Colombia and a bird in Papua New Guinea. Convergent evolution, solving the same lethal problem with the same elegant answer, thousands of miles apart, millions of years apart. The same toxin that can drop a gorilla, cruising through the canopy on a bird no heavier than an orange. Stonefish. In Australian emergency rooms, there's a specific kind of screaming that trauma nurses learn to recognize. Not the sharp cry of a broken bone or the panicked breathing of anaphylaxis. This is different. Patients arrive from the coast begging, not asking, begging, doctors to amputate their leg. Cut it off at the knee, they say. Right now. Just take it. The pain is so absolute, so beyond the reach of any painkiller in the crash cart, that rational adults with no psychiatric history request surgical dismemberment as relief. They're not delirious. They know what they're asking. They mean it. They stepped on a stonefish. The reef stonefish, Sinensea verrucosa, is the most venomous fish in the world, and it is essentially invisible. It sits motionless on the ocean floor for days at a time, covered in a rough, warty skin that accumulates living algae, sediment, and debris until it is physically indistinguishable from a rock. It doesn't look like it's pretending to be a rock. It looks like a rock. Divers have rested their hands on one without knowing. It can also survive outside of water for up to 24 hours, breathing through its thick, moist skin on tidal flats and shallow sandbars. A biological landmine that works on land and sea. Across its back, hidden under fleshy warts, sit 13 dorsal spines made of solid cartilage. Each spine is connected to 2 venom glands at its base, like twin ampules loaded into a hypodermic needle. And here is the mechanical horror. The venom delivery system is not controlled by the fish's nervous system. It's not a decision. It's not a reflex. It's pure hydraulics. The spines are hollow and the glands are pressurized. When your foot comes down on the spine, your own body weight compresses the gland and forces venom up through the channel and into your flesh. The harder you step, the deeper the spine goes, the more venom you receive. You inject yourself. What enters your bloodstream is stonnus toxin, and what it does to human cells is structural annihilation. On contact with red blood cells, the protein creates massive perforations in the cell membrane. Macropores, essentially enormous biological holes punched from the inside. The cell doesn't leak, it ruptures, it explodes outward. Tissue destruction cascades from the entry point through the foot, the ankle, up the leg, through the lymphatic system. The pain isn't a symptom. The pain is the primary weapon. And the stonefish never uses this weapon to hunt. Not once. It catches prey, small fish and shrimp, by sitting still and vacuuming them into its enormous mouth with a rapid suction strike that takes 6 milliseconds. The fastest feeding strike of any vertebrate. The 13 spines of agony on its back evolved for one purpose only: to punish any large, heavy animal foolish enough to step on it. It's a pure defense system, perfected by evolution into the most painful envenomation known to medicine, and it has zero offensive function. In 2019, a British diver named Callum Thomas was wading through shallow sand in Thailand when he stepped on a concealed stonefish. He described the initial sensation as boiling oil being poured directly into his veins. Within minutes, the pain escalated past any frame of reference he had. He lost the ability to speak coherently in the ambulance. Standard painkillers, morphine included, failed completely. At the hospital, doctors submerged his foot in water hot enough to scald skin because heat partially denatures the venom protein, and performed a full nerve block in his leg to stop the pain signals from reaching his brain. It took him months to walk normally again. The stonefish, meanwhile, can sit on a dry rock above the tide line for a full day, breathing calmly through its skin, waiting for the water to come back. Just a rock on the shore. Nothing to see. Pufferfish. Every animal in this series kills you when you reach into its territory. You step in the wrong place, touch the wrong skin, swim through the wrong water. The pufferfish is different. With this one, you sit down at a table, pick up chopsticks, and pay for the privilege. Fugu is the Japanese culinary tradition built around eating pufferfish, specifically species from the family Tetraodontidae, and it requires a kind of training that makes medical school look casual. Aspiring fugu chefs study for years. They take written and practical state examinations. They must demonstrate the ability to cut along exact millimetric lines that separate the edible muscle from the organs saturated with tetrodotoxin. The licensed chefs know that the liver, the ovaries, the gonads, and the skin contain enough concentrated poison to kill everyone in the restaurant. The art, the entire cultural identity of the dish, lies in cutting close enough to the toxic organs that trace amounts of tetrodotoxin reach the meat. Just enough to cause a faint numbing tingle on the diner's lips and tongue. A whisper of death as a flavor. Tetrodotoxin, TTX, is 1,200 times more lethal than cyanide. A single pufferfish carries enough of it to kill roughly 30 adult humans. There is no antidote. In January 1975, one of Japan's most celebrated kabuki actors tested that math. Bando Mitsugoro Waith was a man of extraordinary cultural stature, designated a living national treasure by the Japanese government, a title reserved for the highest masters of traditional art. At a restaurant in Kyoto, he demanded that the chef serve him 4 portions of fugu liver. The liver, the single most restricted organ, the densest reservoir of TTX in the fish's body, banned from commercial service under Japanese law. Mitsugoro claimed he was immune. He had eaten fugu many times. He told the chef he could resist the poison. The chef, overwhelmed by the celebrity and the pressure of refusal, served the liver. Mitsugoro ate all 4 portions. He returned to his hotel. Within hours, he began losing control of his limbs. His voice faded, then disappeared. Tetrodotoxin blocks sodium channels in nerve cells with absolute precision. The muscles that expand the lungs simply stop receiving the signal to move. Mitsugoro died of respiratory paralysis, fully conscious, unable to draw a single breath, alone in his hotel room. The death shocked Japan. It permanently changed the country's fugu licensing and restaurant regulation laws. The evolutionary logic of the pufferfish's toxin is a kind of dark delayed revenge. The poison isn't in the meat. It's concentrated in the organs, the liver, the ovaries, the intestines. The animal is small, slow, and easy to catch. It inflates when threatened, but that's a bluff, not a defense. The real defense activates after the pufferfish is already dead and swallowed. A predator eats the fish whole, digests the organs, and absorbs the TTX. 12 to 18 hours later, the predator dies. The pufferfish is gone, but so is the thing that ate it. Evolution designed a post-mortem retaliation system, a chemical will executed from inside the stomach of the killer. And then there's what happened in Haiti. In the early 1980s, an ethnobotanist named Wade Davis traveled into the rural interior of Haiti to investigate persistent accounts of something Western medicine refused to take seriously: zombies. Not movie zombies. Real people who had been declared clinically dead, buried, and later found walking in remote villages, alive, in a state of profound neurological damage. What Davis documented, and what remains one of the most unsettling intersections of pharmacology and culture ever recorded, was this. Haitian practitioners known as bokors, sorcerers, masters of the dead, prepared a powder from macerated pufferfish, specifically the organs loaded with TTX. This powder was applied to the skin of a victim, usually through a wound or abrasion. The tetrodotoxin entered the bloodstream in a carefully controlled microdose, not enough to kill outright, but enough to suppress respiration and heart rate to levels so faint that trained physicians with stethoscopes could detect no pulse. The victim was declared dead. A certificate was signed. A coffin was sealed. But the victim was conscious, paralyzed beyond the ability to move a single muscle, unable to open their eyes, unable to scream, but aware. They heard the prayers. They felt the weight of the dirt. They were buried alive in full awareness and dug up hours later by the same bokors who'd poisoned them, reanimated in a deep neurological fog, brain damaged from oxygen deprivation, suggestible, stripped of identity and will. They were put to work in the fields. They spoke in fragments. They moved like sleepwalkers. The villages called them zombies. The myth of the Walking Dead wasn't born from superstition. It was built on tetrodotoxin, the same molecule sitting in a pufferfish on a ceramic plate in a Kyoto restaurant, served with a garnish and a warning. The shovels were real, the coffins were real, and the neurotoxin that made it all possible came from a fish that fits in the palm of your hand. But not every killer waits in the ocean or the jungle. Some of them are already inside the walls of your house. Sydney funnel web spider, not in the Amazon, not buried in some uncharted tropical cave. The most dangerous spider alive sits in suburban Sydney, inside a rolled garden hose, underneath a pile of damp leaves by the pool, or tucked deep inside a leather boot left overnight by the back door. The Sydney funnel web spider, Atrax robustus, is a resident of garages, laundry rooms, and backyard sandpits, and it does not behave like any spider you've ever encountered. Corner a house spider and it bolts. Disturb a tarantula and it freezes or flicks irritating hairs. The funnel web does neither. When confronted, it rears back, throws its abdomen into the air, and extends two massive fangs already beaded with venom. And then it walks toward you. Not away, toward. It will actively pursue your foot, your hand, your face. And once those fangs connect, it does not let go. It bites, locks on, and bites again. The mechanics of those fangs explain the damage. Most spiders bite laterally, two tiny pincers squeezing from the sides like tweezers. The funnel web is a mygalomorph. Its chelicerae are oriented vertically, driving downward in vicious overhead chops powered by the full weight of the spider's body. Hospitals across New South Wales have documented hundreds of cases where fangs punched cleanly through leather shoes, denim, and even the hard keratin of human toenails. Gardeners kneeling in soil find out the hard way that this spider treats every barrier as a suggestion. But here's the part that feels almost personal. The funnel web's primary lethal compound, a neurotoxin called delta hexatoxin or robustoxin, is exquisitely calibrated to bind with the sodium channel receptors of only two categories of animal on Earth: insects and primates, not mammals in general. Primates, humans, rhesus monkeys, gorillas. A funnel web bite will send a grown man into cardiac arrest in under an hour. A house cat bitten by the same spider might scratch the spot for a day and forget about it. Dogs barely react at all. The venom is, for all practical purposes, built with our name on it. A biochemical coincidence so specific it feels like a grudge. In 1979, a 4-year-old boy named Markley Henderson was playing in his backyard in Sydney when he was bitten. The delta hexatoxin detonated through his autonomic nervous system in minutes. Within 15 minutes, Markley was salivating uncontrollably, crying thick, viscous tears, and his lungs had begun filling with fluid, acute pulmonary edema in a child barely old enough for school. His heart stopped before he reached the hospital. Markley's death, along with a string of similar tragedies, drove an immunologist named Struan Sutherland into a years-long race to develop a definitive anti-venom. He succeeded in 1981. Since then, not a single confirmed death from a funnel web bite has been recorded in Australia, and the story kept turning. Researchers investigating exactly how the venom kills, specifically how it blocks neural oxygen pathways so rapidly, discovered dozens of unique mini-peptides hiding inside that black fluid. One of them, a protein called Hi1A, demonstrated an astonishing ability to slow ischemic brain damage caused by sudden oxygen deprivation. The same molecular mechanism that drowns a child's lungs in fluid can, when isolated and purified, protect brain cells during a stroke. Clinical trials in Australia are now developing Hi1A as an emergency treatment, something paramedics could administer in the ambulance to buy time before a heart attack patient even reaches the ER. The spider that evolution armed specifically against us may end up saving more human lives than it ever took. Brazilian wandering spider. Its common name in export markets is the banana spider, not because of its color, because the Brazilian wandering spider, Phonutria nigriventer, travels. It nests inside banana bunches in tropical warehouses, survives weeks of refrigerated shipping containers, and arrives in supermarkets across Europe and North America, still very much alive and deeply irritated. It doesn't spin webs. It doesn't sit in corners. This spider is a ground-level predator that actively patrols at night, and when it encounters something larger than itself, it doesn't run. It lifts its two front pairs of legs off the ground, exposing a spread of dark, reddish, urticating hairs underneath, and rises into an unmistakable bipedal threat posture, legs wide, fangs forward, body braced. Then it charges. A spider running at you on two legs with its mouth open is not a sentence anyone expects to be literal. It is. For years, the Guinness World Records listed Phonutria as the most venomous spider on Earth. The toxin cocktail, primarily PNTX2/6 and PHTX3, saturates synaptic clefts and floods nitric oxide pathways, creating simultaneous cardiac chaos, serotonin inhibition that suppresses breathing, and total neuromuscular hyperexcitation without an off switch. As little as 0.06 milligrams can send a small mammal into fulminant paralytic collapse in minutes. The body doesn't shut down, it revs until it breaks. And then there's the other symptom. In male bite victims, The venom reliably causes priapism, prolonged, agonizingly painful erections that can last for hours. A man in anaphylactic shock, struggling to breathe, heart rate destabilizing, while simultaneously experiencing an uncontrollable and excruciating erection in a hospital emergency room. It is medically documented, pharmacologically understood, and still somehow one of the most absurd sentences in all of toxicology. In 2014, a family in Staffordshire, England, bought a bag of imported Brazilian bananas from a local supermarket. At home, the mother opened the bag and found a fully grown wandering spider that had traveled thousands of miles in a climate-controlled container. The spider immediately assumed its armed bipedal posture, slapping its front legs through the air. The family evacuated the house. The entire residence had to be sealed, quarantined, and professionally fumigated before they could return. But the priapism turned out to be the important part. Researchers at the Federal University of Minas Gerais in Brazil isolated the exact molecule responsible, PNTX2R6, and reverse-engineered its nitric oxide pathway. From that single peptide, they synthesized a compound called BZ371. In laboratory trials, BZ371 outperformed the most famous synthetic erectile dysfunction treatment on the planet, the one that generates billions of dollars annually, with fewer side effects and a more targeted mechanism of action. A spider whose bite can kill you in under 2 hours also accidentally invented a better version of the world's most profitable pharmaceutical product. The venom giveth and the venom taketh away. Deathstalker scorpion, $39 million per gallon. That's the current estimated market value of the venom produced by the Deathstalker scorpion, Leiurus quinquestriatus. It is the most expensive liquid on Earth, not per ounce, per gallon. More valuable than printer ink, king cobra venom, and human blood combined. There is an ancient evolutionary rule among scorpions, and it holds with remarkable consistency. The bigger the pincers, the weaker the venom. Species with massive, crushing claws rely on brute mechanical force to subdue prey. They barely need chemistry. The Deathstalker has pincers so thin and fragile they look ornamental. Pale, slender tweezers that couldn't crack a grain of rice. Every ounce of survival capital that didn't go into those pincers went directly into the tail. That tail, the telson, the swollen bulb tipped with a curved spike, is a precision instrument. Independent muscular sensors allow the scorpion to calibrate each sting individually. It can deliver a bland, metabolically cheap, dry sting, pure pain, no real venom expenditure, as a warning shot. And milliseconds later, if the threat persists, it dumps the full cocktail of agitoxins directly into the wound. The economy of violence is deliberate. Venom is expensive to produce. The Deathstalker doesn't waste a drop it doesn't have to. When cornered, though, economy goes out the window. Its reaction speed is borderline ballistic. The stinger whips forward in an arc over the cephalothorax at roughly 130 centimeters of linear swing, delivering rapid successive injection strikes in a frenzy of defensive panic. It doesn't sting once. It hammers. In Upper Egypt, a 6-year-old girl was stung in her own bed. The scorpion had sheltered inside her bed sheets during the day and struck when she climbed in at night. Within the hour, she presented extreme hypertension, severe cardiac arrhythmia, and pulmonary edema, her heart flooding itself as the agitoxins dismantled autonomic control. Doctors placed her on mechanical ventilation and administered triple doses of imported antivenom before her myocardium stabilized. She survived. Many children in rural North Africa and the Middle East, where antivenom is scarce, do not. And then there's the reason for the price tag. A pediatric oncologist named Jim Olson was studying the Deathstalker's venom when his team isolated a purified peptide called chlorotoxin. It does something no synthetic drug has been able to replicate. In the human body, chlorotoxin drifts harmlessly past every healthy cell it encounters. Tissue, blood vessels, neurons, organs, touching nothing. But the moment it contacts a glioma cell, the aggressive, invasive tissue of brain cancer, it binds, immediately, irreversibly. It finds the cracks in destructive tumor cells with a precision that borders on impossible. Olson's team attached a faintly fluorescent marker to the peptide. Under surgical lighting, the tagged chlorotoxin causes brain tumors to glow. Surgeons operating in the delicate darkness of a craniotomy can now see exactly where healthy tissue ends and cancer begins. In real time, at the cellular level. They called it tumor paint. $39 million a gallon sounds obscene until you're the person on the operating table with a glioma, and the only thing standing between a clean resection and permanent brain damage is a molecule that came out of a scorpion the size of your thumb. Russell's viper. 40,000 confirmed deaths per year. Some estimates push it past 50,000. The Russell's viper, Daboia russellii, kills more human beings annually than any other snake on Earth, and it does it in the most ordinary place imaginable: rice paddies. Across India, Sri Lanka, Bangladesh, and Myanmar, barefoot farmers step on this snake every single day. It is the heaviest entry in this part for a reason. It doesn't rattle. It doesn't have a rattle. What it produces instead is a hiss, the full forced exhalation of oversized lungs pushing air through constricted membranes that herpetologists have ranked as the loudest, most explosive, and most psychologically disturbing warning sound of any snake alive. If you've heard it described as a pressure valve releasing, that's close. If you've heard it in person, the description doesn't matter anymore. The venom itself is a complex of dozens of destructive proteins, but its most insidious target is one most people have never heard of: the pituitary gland, a pea-sized structure at the base of the brain that functions as the master regulator of human hormones. Growth, metabolism, reproduction, stress response, all of it runs through the pituitary. Russell's viper venom attacks it directly, causing hemorrhagic infarction in the gland's delicate tissue, while simultaneously triggering renal failure, massive edema, and catastrophic shock throughout the rest of the body. And here is the cruelest part. Surviving a Russell's viper bite is not the end of the story. For many victims, it is the beginning of something they describe as worse. The condition is called Sheehan syndrome, hypopituitarism caused by venom-induced destruction of the pituitary. In male survivors, the effects emerge over weeks and months with devastating clarity. Healthy young farmers who walked into the hospital and walked out alive began losing their body hair. Beards stopped growing. Muscle mass eroded. Genital organs underwent irreversible atrophy and retraction. Sex drive vanished completely. Voices thinned. Dense tissue wasted away. Vigorous men in their 20s and 30s were biologically regressed. Their endocrine systems collapsed into states resembling prepubescence. Not aging, reversal. A 28-year-old farmer in Sri Lanka was bitten while working his field. Anti-venom saved his life. In the months that followed, his beard stopped growing. His voice changed. His muscles withered despite eating the same meals, doing the same work. An MRI revealed a hemorrhagic infarction had destroyed his pituitary gland. The diagnosis was permanent chemical and hormonal castration. He will require hormone replacement therapy for the rest of his life to maintain basic adult biological function. Among Asia's Big Four, the quartet of snakes responsible for the overwhelming majority of snakebite mortality across the continent, Russell's viper consistently leads the count: 40 to 50,000 dead per year, mostly farmers, mostly poor, mostly barefoot in a flooded field at dawn. But not every deadly animal announces itself with fangs bared and a hiss you can hear across a field. Some of the most dangerous creatures on Earth were underestimated, sometimes for decades, because they simply don't look like killers. Boomslang. In September of 1957, a herpetologist named Carl Patterson Schmidt sat at his desk at the Field Museum of Chicago and began writing. Not a paper, not a letter, a diary. He was documenting, hour by hour, the systematic shutdown of his own body. He knew exactly what was happening. He just didn't believe it could. Earlier that afternoon, a colleague had brought him a juvenile snake for identification, a slender, bright-eyed thing from sub-Saharan Africa. A boomslang, Dysphylitis typus. The small snake bit Schmidt on the thumb during handling. A minor nip, barely bled. Schmidt looked at the wound, looked at the snake, and made a decision that would cost him everything. He sent the colleague away, declined medical attention, and went back to work. He had good reason, or thought he did. European biology textbooks at the time classified the boomslang as shy and essentially harmless to humans. It's an opistoglyph, a rear-fanged snake. Its venom-delivering teeth sit far back in the jaw, behind rows of smaller teeth, which means a bite on a large-limbed primate like a human rarely achieves deep enough penetration to inject a meaningful dose. That was the logic. For decades, safari guides repeated it. Zoology professors taught it. Schmidt believed it. But belief doesn't neutralize hemotoxin. The boomslang's venom yield per bite is almost laughably small, between 1.6 and 8 milligrams. A bee injects more fluid. But the LD50 of this particular hemotoxin cocktail is so violently low that just 0.1 milligrams in the bloodstream is enough to trigger hemorrhagic shock in a grown adult. You don't need volume when the chemistry is that precise. What the venom does is quietly catastrophic. It targets prothrombin, the protein your liver manufactures so your blood can clot. The toxin prematurely activates it, not at the wound, everywhere. Across every capillary, every vessel, every organ simultaneously. The body, confused by thousands of false alarm signals, burns through its entire reserve of clotting factors, forming useless microclots in places where no injury exists. Doctors call it disseminated intravascular coagulation, DIC. The name sounds clinical. What it means is that your blood uses up everything it has trying to patch wounds that aren't there. And when the liver's protein factories hit zero, when every last molecule of coagulant has been wasted on phantom wounds, the blood simply stops binding. It can't clot anymore, anywhere, for any reason. It flows like thin red water. From the gums, from the nose, from the tear ducts. The victim cries blood before they understand what's happening to them. Schmidt didn't know any of this yet, but he was about to learn it from the inside. At 4 p.m., he noted the first symptom, chills. A slight shaking he couldn't attribute to the room temperature. He wrote it down. By 9 p.m., he was bleeding from his mouth and nose. Steady, unprovoked bleeding. No trauma, no irritation. Just blood appearing where it shouldn't be. He recorded this too, with clinical detachment. At 5:30 a.m. the next morning, he woke, nauseous. He vomited. He ate a light breakfast anyway and sat down to write again. At 7 a.m., his entry read, low fever, continuous bleeding from mouth and nose, but no pain at the finger site. No pain at the bite. That's the boomslang's cruelest trick. The bite doesn't hurt. The victim sleeps, and the hemorrhage quietly becomes irreversible overnight. By 10 a.m., Schmidt was urinating pure blood. His platelets were being annihilated faster than his marrow could produce them. The DIC had entered its terminal phase. Every vessel in his body was now leaking freely. By noon, he was coughing blood, not streaks, mouthfuls. His lungs were filling with fluid his body could no longer contain. When the ambulance finally arrived, there was almost nothing left to save. Generalized internal hemorrhage had liquefied the integrity of his organs. Carl Patterson Schmidt was pronounced dead at 1 p.m. on September 26, 1957, leaving behind the coldest, most meticulous first-person account of an envenomation in the history of science. His handwriting in the final entry was trembling. When threatened, the boomslang doesn't strike first. It inflates. It forces air into its neck and trachea until the skin balloons to grotesque proportions, doubling its diameter under the leaves, faking the wide flared hood of a giant enraged cobra. It's a performance, a bluff. It doesn't belong to that family. It just learned to look like one to avoid wasting the few milligrams of venom that can quietly rewrite a man's final hours. And for decades after Schmidt's death, some textbooks still called it harmless. Slow loris. There's a gesture the slow loris, Nycticebus, makes that has broken the internet more than once. It raises its small, round arms above its head, slowly, gently, like a toddler asking to be picked up. Tickle videos went viral. People shared them with heart emojis. What nobody told them is that the animal wasn't surrendering. It was arming its mouth. The slow loris is the only venomous primate ever discovered. That sentence should bother you more than it does, because it breaks a pattern that evolution held for tens of millions of years. Venom, as a biological strategy, is almost exclusively the domain of simple organisms, jellyfish, cone snails, spiders, creatures with limited intelligence that need chemical weapons to compensate for what they lack in cognition. Complex binocular vision, mammalian brains, social intelligence, our primate lineage traded all of that for other survival tools. Venom was supposed to be off the table. The slow loris didn't get that memo. On the inside of its elbows, tucked into the crease where the arm bends, sits a small secretory patch called the brachial gland. It produces a dense, sharp-smelling chemical oil. On its own, exposed to air, the secretion is inert, harmless. But when the loris raises its arms, that adorable viral gesture, it brings those glands to its mouth. Its tongue works the oil into its saliva. And in seconds, two individually harmless secretions combine into something new. A volatile toxin that melts proteins on contact. That's the alchemy. The arms go up, the tongue goes to the elbows, and the next bite carries a payload that no other primate on Earth can deliver. But the strangest part isn't the venom itself. It's what the venom is made of. Brian Fry and a team at the University of Queensland ran genetic sequencing on the toxin molecule and found something nobody expected. The lethal protein secreted from the loris's brachial glands and activated by its saliva is geometrically near identical to fel D1, the protein found in the saliva and skin of ordinary domestic house cats. The same molecule that makes millions of people sneeze, itch, and wheeze around their pets. The slow loris didn't invent a new weapon. Evolution took an existing feline allergen, one that causes mild discomfort in most humans, and concentrated it, weaponized it, supercharged it to levels that trigger full anaphylactic shock in mammalian predators. The victim's throat swells, the glottis closes, breathing stops, not from a neurotoxin, not from a hemotoxin, from an allergic reaction so severe that the immune system kills the host trying to fight it. Death by cat dander, concentrated 1,000-fold. A Western researcher at an ecological rehabilitation center in Indonesia learned this firsthand. He was handling a rescued slow loris, a trafficking survivor, confiscated from a dealer who had already pulled its teeth. But this one still had them. The animal calmly raised its arms, licked its elbows with deliberate care, and delivered a single deep bite to the webbing between his thumb and index finger. The researcher barely flinched. He'd handled primates for years. 2 minutes later, his lips were tingling. His tongue felt thick. His throat began to tighten. Not from fear, but from tissue swelling shut from the inside. He collapsed in full respiratory arrest before he could walk 30 meters to the medical station. He survived only because the rehabilitation team had emergency epinephrine on site and recognized anaphylaxis when they saw it. The bite wound didn't heal cleanly. The tissue around the puncture necrotized over the following weeks. The skin blackened, peeled, and took months of treatment to close. He was left with deep, permanent scars on his dominant hand. He got lucky. The animals don't. Every single slow loris sold through the Asian black market has had its teeth pulled before sale. Every one. Criminal traffickers use wire cutters or pliers. No anesthesia, no antiseptic. They wrench out the sharp incisor teeth, the ones that deliver the venom, because a buyer paying thousands of dollars for an exotic pet in a London flat or a Tokyo apartment doesn't want to learn about anaphylaxis the hard way. Many of the animals die from infection or shock before they ever reach a customer. The ones that survive can't eat properly for the rest of their shortened lives. The tickle videos keep going viral. The arms keep going up. Komodo dragon. Here's something almost every nature documentary told you in the 1990s. The Komodo dragon, Varanus komodoensis, kills with bacteria. Its mouth, they said, was a biological cesspool, a warm, rotting incubator of dozens of fatal bacterial strains living in the shreds of decomposing meat between its teeth. A single bite would infect the wound. The prey would wander off and die of sepsis days later. The dragon just followed the smell. It was a satisfying story, visceral, easy to animate. And it was wrong. In 2009, a biologist named Brian Fry, the same researcher who decoded the slow loris, put a preserved Komodo skull under an MRI scanner. He wasn't looking for bacteria. He was looking for structure. And what the cold fluorescent imaging revealed wasn't rotting tissue or microbial colonies packed into the gaps of its teeth. It was glands. 6 enormous spongy pouches of glandular tissue hidden in the maxillary bones, connected by capillary ducts to openings along the serrated edges of the teeth. The Komodo dragon doesn't kill with infection, it kills with venom. It always did. The toxin proteins do several things at once. They dismantle the fibrin scaffolding that allows blood to clot. They force capillary vessels to dilate far beyond normal capacity, dropping blood pressure catastrophically. They ensure that every wound, no matter how small, bleeds freely, without resistance, without end. The result isn't sepsis, it's controlled hemorrhagic collapse, a slow, mechanical draining. And the dragon knows exactly how to use it. The hunt doesn't look like a hunt. A Komodo dragon will wait for hours in low foliage near a game trail or a shallow swamp, the kind of murky, knee-deep water where a 500-kilogram water buffalo has to slow its stride. When the moment comes, the dragon doesn't lunge for the throat. It doesn't try to wrestle an animal that outweighs it 5 to 1. It delivers a single, precise slashing strike to the back of the legs, hamstrings, tendons, the curved joint behind the knee. One cut, maybe 2, deep enough to open the tissue wide, deep enough for the venom to seep into the bloodstream. Then it stops. This is where it gets unsettling. The dragon doesn't press the attack. It doesn't chase. It backs off to a comfortable distance, settles into the grass, and sits down. It watches. For hours, sometimes for 2 or 3 days. It follows the blood trail at a casual walk, a patient, silent observer trailing the exact drip pattern through thin puddles, across cracked mud, into the tree line. The buffalo keeps moving because it doesn't know it's already dead. But the venom keeps the wounds from clotting, the blood keeps draining, and the animal's pressure keeps dropping until the legs finally buckle and the massive body folds into the dust. Only then does the dragon move in to feed. No struggle, no second attack, just patience and chemistry. Here's the paradox that makes the whole thing even stranger. Biomechanical analysis of the Komodo's jaw, that massive, terrifying skull, reveals a bite force that is, objectively, embarrassing. Pound for pound, a Komodo dragon bites with less force than a house cat, less than a turkey. That enormous head isn't built for crushing, it's built for slicing. The teeth are laterally compressed, serrated like steak knives, set in a jaw designed for one motion, a fast, pulling slash that opens wide, shallow wounds across as much surface area as possible. Maximum exposure, minimum effort. Phil Bronstein found this out in 2001.
Bronstein, a newspaper editor and at the time husband of actress Sharon Stone, was visiting the Los Angeles Zoo when he was offered the chance to enter a Komodo dragon enclosure for an up-close experience. The reptile moved fast and low, clamped onto his bare foot, and shredded the tendons with its serrated teeth before handlers could intervene. Bronstein escaped the enclosure, but the damage was already compounding.
Venom from the submandibular glands entered the wound and triggered massive bleeding that doctors struggled to control. He required extensive reconstructive surgeries and intensive treatment to reverse the cascading blood pressure collapse the venom had set in motion. A 3-meter lizard with the bite force of a pet cat. It didn't need to crush anything. It never did. But not every lethal animal bothers with venom, or teeth, or even moving at all. The last 3 on this list come from the ocean and from places nobody thinks to look, and they are by far the strangest. Dubois sea snake. The most venomous snake in all the world's oceans isn't a cobra. It isn't a mamba that learned to swim. It's a quiet, slate-gray serpent called the Dubois sea snake, Iapysurus duboisii, drifting through the warm reefs of the Coral Sea and the Timor Sea with an LD50 of 0.044 milligrams per kilogram. For context, that makes it roughly 10 times more potent than most land cobras. Drop for drop, nothing in the ocean carries a deadlier bite, and it barely needs to breathe. Most sea snakes have to surface every 30 minutes or so to fill their lungs. The Dubois doesn't rush. Its skin, the actual scaled surface of its body, can absorb dissolved oxygen directly from cold, deep water. It breathes through its hide, which means it can stay down longer, hunt deeper, and drift through the dark thermocline where other sea snakes can't follow. What happens when it bites is a specific kind of catastrophe. The venom carries a potent myotoxin, a molecule that targets skeletal muscle fibers and tears them apart at the cellular level. The technical term is rhabdomyolysis, and what it means in practice is that your muscles dissolve. Not metaphorically. The protein filaments that make up muscle tissue shatter into fragments, releasing a flood of cellular debris, myoglobin, potassium, phosphate, directly into the bloodstream. The kidneys try to filter this wave of toxic waste. They fail. The urine turns black, not dark, not brown. Black, like ink, choked with the destroyed remnants of muscle that should never have entered the renal system. Then the kidneys shut down entirely. Here's where the story turns absurd. The emperor of all lethal seas has microscopic fangs, 1.8 millimeters, less than the thickness of a nickel. Tiny, fragile, hollow needles of bone so short and so weak that the snake routinely fails to penetrate anything thicker than bare skin. It strikes a wetsuit and bounces off. It bites a diver's glove and slides away, frustrated, having delivered exactly nothing. A single layer of thin neoprene, the cheapest, flimsiest dive gear on the market, is a complete and total shield against the most venomous sea snake on Earth, which makes you wonder how anyone ever dies from one. A young fisherman in the Timor Sea found out during a night trawl. The nets came up heavy with fish, and tangled among them were dozens of Dubois sea snakes, common bycatch in those waters. He reached in barehanded to sort the catch. One of the snakes scratched his finger, not bit, scratched. The puncture was barely a millimeter deep. It hardly bled. He shook his hand, wiped it on his pants, and kept working. 2 hours later, his forearms started to cramp, then his thighs. The cramps deepened into a spreading, grinding ache that didn't respond to stretching or rest, because it wasn't a cramp. It was his muscle tissue coming apart. His urine turned black. He collapsed on the deck, paralyzed from the chest down, fully conscious, staring at the sky while his kidneys processed the last toxic flood they'd ever handle. The boat was hours from shore. He died of acute renal failure before anyone could get anti-venom into his bloodstream, all from a scratch he didn't think was worth mentioning. But the strangest part of the Dubois sea snake isn't its venom or its useless fangs. It's why it bites divers at all. For decades, researchers documented aggressive strikes on scuba divers, sudden, unprovoked lunges in deep water. The snake would wrap itself around a flipper, coil against a leg, and bite repeatedly with what looked like genuine fury. Every report described the same thing. The snake seemed enraged. It wasn't. Marine biologists eventually proved that the attacks happened almost exclusively during mating season, in deep water, in low visibility. The males, essentially blind in the dark, were searching for females by shape and texture, and what they found instead were large, smooth, dark, rubbery objects gliding slowly through their territory. Dive fins, neoprene legs, wetsuit boots. The Dubois sea snake wasn't attacking divers. It was trying to mate with them. Every furious, venomous lunge was a love bite. Every coiling embrace around a flipper was a courtship display. The most venomous creature in the ocean was hugging the wrong partner in the dark and getting increasingly desperate about it. Gila monster. There's a sound that people who've been bitten by a Gila monster, Heloderma suspectum, never forget. It's not a hiss. It's not a snap. It's the wet grinding of jaw against flesh. A slow, rotational crunch, like someone chewing tough meat with their mouth open. Because that's exactly what's happening. The Gila monster doesn't strike and release. It bites and holds, and then it chews. Most venomous animals have elegant delivery systems. Snakes have hollow hypodermic fangs, clean, precise, spring-loaded syringes that inject venom in a fraction of a second. The Gila monster has none of that. Its venom glands sit in the lower jaw, not the upper. The teeth aren't hollow needles, they're grooved. Shallow capillary channels run along the outer surface of each tooth like tiny gutters, and the venom doesn't inject. It oozes, it pools in the gums, mixes with saliva into a foamy, toxic drool, and seeps upward through the grooves by surface tension and gravity. But only if the teeth are buried deep enough in flesh for the channels to make contact with open tissue. Which means the Gila monster has to chew. It clamps down, locks its jaw, digs its claws into whatever it's gripping, and begins to roll. A slow, deliberate, twisting rotation, torquing its entire body to grind its teeth deeper, tearing the wound wider, working the grooved teeth back and forth through the lacerated flesh to channel venom into every exposed nerve ending. It doesn't stop. It doesn't flinch. It doesn't let go. It keeps grinding, rolling, chewing, sometimes for 5 minutes, sometimes for 15, until it has physically exhausted itself or until someone pries its jaw open with a tool. And here's the part that makes the whole thing absurd. This entire elaborate, grotesque medieval machine exists to eat eggs. The Gila monster doesn't chase prey. It doesn't ambush rodents or stalk birds. It waddles slowly through the Sonoran desert, finds an unguarded nest, and eats the eggs inside. Still, silent, defenseless eggs buried in sand. That's its primary food source. This animal evolved the most labor-intensive venom delivery system in the reptile kingdom to rob cradles. The venom itself is genuinely dangerous, a cocktail of toxins that causes immediate, overwhelming pain, followed by rapid drops in blood pressure, nausea, and in severe cases, respiratory collapse. But the delivery is so crude, so dependent on sustained chewing and tissue contact, that it feels like a biological rough draft. A prototype that evolution never bothered to refine, because it didn't need to. The eggs weren't fighting back. In February 2024, a man named Christopher Ward found out that the prototype still works. Ward was 34, lived in Colorado, and kept 2 Gila monsters illegally in his apartment. One of the animals bit his hand and initiated the full sequence: jaw lock, claw grip, twist and grind. It stayed clamped to his flesh for several minutes before someone managed to lever its jaw open. Ward began vomiting almost immediately. He lost consciousness shortly after. By the time paramedics got him to the hospital, he was in acute respiratory failure. He was placed in the ICU on life support. Days later, doctors declared brain death. The first confirmed Gila monster fatality in the United States in nearly 100 years. February 2024. Not a historical footnote. Last year, platypus, a duck-billed, beaver-tailed, egg-laying mammal that navigates underwater by detecting the electrical fields of shrimp. If that sentence sounds like it belongs on a different list, that's the point. The platypus, Ornithorhynchus anatinus, is the last animal here, and it earned the spot. The weapon isn't in its mouth. It's on its ankles. Males carry a curved, hollow spur on each hind leg, about 1.5 centimeters long, tucked against the inner shin, hidden in a fold of cartilage and fur. It doesn't bite. It kicks. The spur connects directly to a pair of crural glands buried deep in the thighs, and when the platypus drives the spur into flesh, pelvic muscle contractions pump venom through the hollow spike like a pressurized syringe. What comes out is unlike any other venom on this list.
Platypus venom doesn't cause necrosis. It doesn't dissolve muscle. It doesn't shut down the heart or paralyze the diaphragm. It does something that might be worse. It reprograms pain. The venom contains a class of hyperalgesic peptides, molecules that hijack the peripheral nervous system and crank nociceptive signaling to maximum. Every nerve ending in the affected area begins firing at full intensity, continuously, in response to stimuli that should be harmless.
A light touch feels like a crushing grip. Cool air feels like an open flame. The brush of a bedsheet against the skin registers as a burn. The pain isn't proportional to the injury. The pain is the injury. A neurological rewrite that turns the body's own sensory system into a torture device, and it lasts. Australian military veterans, men who survived combat in Vietnam, who'd been shot, who'd walked through jungle for weeks without boots, have described platypus stings as the worst pain of their lives. Not in the moment. For months. Documented cases show uninterrupted, generalized agony lasting 4 full months without a single day of relief. In emergency wards, doctors administered the heaviest morphine protocols available. Continuous intravenous drip, escalating doses, arterial delivery routes designed for terminal cancer patients. The morphine did nothing. The venom's mechanism bypasses opioid receptors entirely. The most powerful painkiller in the medical arsenal, pumped directly into the bloodstream at doses that would sedate a horse, and the patients kept screaming. Morphine did nothing. The spur exists because of sex. During the Australian winter breeding season, Male platypuses enter a hormonal frenzy. They seek out other males, wrestle in the frigid shallows of rivers and creeks, and drive their venomous spurs into each other's bodies, repeatedly, mercilessly, fighting over nesting territory or a single receptive female. These are not dominance displays. These are full-contact, venom-loaded brawls between small, furry animals that look like they were designed by a committee that couldn't agree on anything. And the platypus is, in the deepest sense, an anomaly. It's a venomous mammal. The entire class Mammalia, every primate, every cat, every whale, every bear, every bat, every rodent on the surface of the earth, abandoned venom somewhere in the deep evolutionary past. The genetic machinery was discarded or never developed. No dog has venom glands, no horse, no human. The platypus is the lone holdout, carrying a lethal weapon that its entire taxonomic class gave up millions of years ago, like an animal from a draft of life that was never supposed to ship. But here's the part that changes the story entirely. In the early 2000s, researchers analyzing the biochemical composition of platypus venom found something that had no business being there. Mixed in among the pain peptides and the defensive toxins, buried in the secretions of a reproductive combat weapon, was a hormone called GLP-1, glucagon-like peptide 1, the same hormone produced by the human pancreas to regulate blood sugar, the same molecule that signals insulin release after a meal. It was sitting in the venom of a duck-billed creature's ankle spur, being pumped into rival males during mating fights in frozen Australian rivers. That discovery cracked open an entire field of research. Scientists realized that the platypus version of GLP-1 was far more stable and longer-lasting than the human form, resistant to the enzymes that normally break it down in seconds. That stability became the blueprint. Pharmaceutical researchers used it to develop a new class of synthetic GLP-1 receptor agonists, drugs that mimic what the platypus venom does to glucose regulation, but without the 4 months of screaming. Those drugs became the most prescribed diabetes medications on the planet. The injection pens used by millions of people worldwide to manage type 2 diabetes trace their biochemical lineage directly back to the venom gland of an animal that looks like it was assembled from spare parts. The deadliest thing on this list didn't just kill, it treated a disease that affects 500 million people. And it did it with the same molecule it uses to fight over a mate in a cold river at the bottom of the world.